All right, so this lecture is going to be a little bit different than some of our previous, not in the way it's structured, but in what we're going to do with it. And so um, this lecture really is just a crash course in what we call the central dogma of molecular biology. And then on Friday, we'll have a short amount of time where you can ask questions over the video over those concepts, and then we're going to dig deeper on Friday. We're going to take it to the next level. We're not going to spend a great deal of time hitting the same concepts covered in this video, but instead we're going to focus on some of the more background elements of what's going on here. So a few key terms. First term, the central dogma of molecular biology. And what this is basically the idea that a gene exists so that it can be transcribed into RNA and then translated into protein. As the idea became very popular in the 1950s and 1960s following the description of the structure of DNA as we started to understand a great deal more about how it works and what it does. And this was kind of the idea that this is what genes do, and this is the only thing genes do, is that they get transcribed into RNA and then translated into protein. We now know that most of the portions of DNA that get transcribed into RNA never get translated into protein, at least in the human genome. Initiation, this is found in both transcription and in translation. And so the process of DNA being written into RNA is transcription, RNA being becoming protein is translation. Both of those have an initiation step in which the machinery that work in those processes are the machinery is recruited, the machinery is assembled, and the process begins. Right after the process begins, you shift over to elongation, again in both transcription and in translation, and this is going to be by far the longest step in both of those processes. Elongation ends with termination in both transcription and translation, and in this part, basically the machinery falls apart. The machinery falls apart, releasing the DNA that is being transcribed into RNA. It releases that RNA, or in the case of translation, the machinery falls apart, releases the RNA that is being translated into protein, which also gets released, or at least translated into a polypeptide chain that can then become a protein. Next one is splicing. So in between translation and in between transcription and translation rather, there are 3 steps of RNA modification. One of those is splicing, in which portions of that RNA molecule are removed from the strand prior to the RNA leaving the nucleus and being translated into a polypeptide chain. Codons. Codons are words constructed from 3 nucleotides, and every 3 nucleotides is a codon, and then these codons are read essentially as tRNA lines up its anticodon to match that codon. And so tRNA is a strand of RNA in the same way mRNA is a strand of RNA, and the tRNAs have an anticodon made up of 3 nucleotides, and when those 3 nucleotides can form hydrogen bonds with the 3 nucleotides in the codon, the proper tRNA is in place, and it can move its amino acid onto the growing polypeptide chain. Charging. Charging is a process we're not really going to see at all in this video, but we will definitely talk about on Friday. It is the process of attaching an amino acid to a tRNA that the tRNA can bring into this translation machinery to facilitate growing the polypeptide chain. All right, so initiation of transcription. First thing that happens is RNA polymerase binds about 50 base pairs above or upstream of the site of transcription. This is called the promoter region, and it forms what's called a closed complex. Now, unlike DNA polymerase, RNA polymerase is self-priming. It can attach a nucleotide where there is no nucleotide to attach it to, other than, of course, on the template strand, but not on this new strand. It is self-priming. So RNA polymerase then opens up and forms an open complex. As it moves downstream, it gets about 17 base pairs upstream of the site of transcription, and then it opens it up and continues to move downstream. Now, the initiation site is basically the site at which the first RNA nucleotide is added, and this is basically at site 1. Now, RNA polymerase writes a short little 10 to 20 base pair long RNA transcript that then is removed, and then elongation takes over. And so elongation, once now RNA polymerase is fully cleared the promoter region, it about 23 base pairs upstream from the focal site, the gene, and from there just moves along the DNA, reading the template strand and writing a complementary RNA strand. So newest nucleotide is always added at the 3' end. So like DNA polymerase, RNA polymerase can only write in the 5' to 3' direction. That's an important thing to keep in mind as we move forward. Now, these newly synthesized bases, this new strand of RNA, actually binds to the DNA as it's being written. Now, it doesn't stay bound to the DNA. You only bind about 8 base pairs of it, creating this DNA-RNA hybrid, and that helps to stabilize this process. It helps to keep RNA polymerase attached to the template strand, and it helps to keep the RNA from just floating around in the nucleus where it can be degraded by machinery there. So unlike DNA polymerase, at least DNA polymerase 3 and DNA polymerase 1 that have this 3' to 5' exonuclease activity where they can back up and then write the new bases, RNA polymerase has very limited abilities to repair the strand if it adds the wrong nucleotide in a particular place. So elongation will continue all the way up until the point at which we reach termination. And at termination, it works a little bit differently in prokaryotes and eukaryotes. We'll talk about that more on Friday. But here, one type of termination basically works this way, in that the RNA basically it will keep growing, and then at the very end of where the DNA is being read and needs to be transcribed, you will transcribe a section of RNA that becomes what we call palindromic. And so the DNA sequence has this palindromic sequence, and then the RNA does as well. So you have 8 consecutive bases there where they are G, CCG, C, GGC. And so what that does is when you transcribe the RNA into that, you get those complementary bases lining up, and it creates this. Oh, let me get the pen, and we are going to use a different color other than red. Let's use blue, and I don't know, can we make it any bigger? I think that might be as big as we can make it. All right. So you create this structure here that basically distorts the ability of RNA polymerase to remain attached to the template strand. It tugs on it, if you will, causing the RNA polymerase to fall off the DNA, releasing the DNA and releasing the RNA transcript. So again, in between transcription and translation, there are 3 steps of RNA modification. So you have right here, we've got what's called the pre-mRNA, and then here we've got, closer to getting to that mature mRNA, but not completely mature yet. So here you can see that we've got that part of that promoter region still as part of the beginning of our RNA transcript. So that needs to get cut off. We'll cut that off. There are machines that will function to cut that off there. And then there are all of these little sections of this called introns. And those introns need to be spliced out. So there's a complex called the spliceosome, and we'll talk more about this on Friday, that functions to recognize which portions of these are introns and cut them off, which shortens the overall length. We also need to add what's called a 5' guanine cap, which will be a cap added here that is a guanine nucleotide, a guanosine. It's an RNA nucleotide that's attached backwards. And so what that cap functions to do, and we'll see this on Friday when we talk in more detail about translation, is it actually helps to inform the ribosome where to bind onto this RNA molecule, and it also provides a little bit of energy for that process. So this is attached to what's called the 5' untranslated region. So here's this right here, this 5' untranslated region, and there we add this 5' guanine cap. We also, the third thing, so we've got splicing, cutting these introns out here. We've got adding the 5' guanine cap. We also need to add a poly A tail to the 3' untranslated region, which is right there. And so that's going to function to do a couple of things. One is it allows the RNA to leave the nucleus. Two is it protects the end of the RNA, part that needs to be translated from disappearing. So in the cytosol, there are nucleosomes that will break down RNA, and so the RNA will not last forever inside of the cytosol. And so this poly A tail is a really long portion of the RNA that can be chewed down before you start to lose some of the material that needs to be translated. So in some ways it functions analogous to the telomere on a chromosome. All right, so now we've written our RNA transcript, we've modified and matured our RNA, and now we can translate it into protein. Oh, sorry, before that, I just wanted to mention what's called alternative splicing, in that sometimes a single gene can actually produce multiple RNA products. And the way that this happens is that we can cut or splice this RNA transcript in several different ways. And so over here, we've got a nice illustration of this. So here's a gene where you've got basically these three portions of here. And then, so here are cutting out the introns in between 1 and 2, and then cutting out 3 as well. And so it only then includes these regions 1 and 2. Here's an alternative way we can splice it, where we can cut from here to here and from here to here, producing this product here. And so the same RNA transcript can be cut in multiple ways to produce different mature RNAs. Same thing is illustrated on this side here, where we've got, in this case, we are cutting out 8, cutting out that region, and that gives us just 7, 9, and 10. And then here, we actually cut everything before 8 and cut this little portion here. And so we got 8 and a portion of 9 and then 10. Again, different ways we can cut the pre-mRNA to produce different mature RNAs. And different mature RNAs are going to be translated into different polypeptides. Now, finally, we can talk about translation. And so I mentioned earlier that both transcription and translation begin with an initiation step. And so an initiation of translation, we have to assemble all of our machinery necessary for carrying out translation. So first thing that happens is we've got this mRNA, this mature mRNA that will bind to the 3S, the small ribosomal subunit, and it'll bind being facilitated by several initiation factors as well as being energized by hydrolyzing GTP. And then this small subunit will find what's called the start codon. And again, remember these codons are words formed by 3 nucleotides. And so the start codon is AUG. That particular sequence always functions as the start codon. And we have a tRNA here that has the anticodon UAC that binds to the start codon. And the amino acid that this tRNA has been charged with is methionine. And so the first amino acid in every polypeptide formed is methionine. Now, that doesn't mean that the first amino acid in every functional protein is going to be methionine, because the beginning portion, what you call the C terminus of this protein, may end up getting cut off, but at least every polypeptide translated begins with methionine. Once the first tRNA binds to this start codon here, we recruit and attach the large ribosomal subunit, the 50S subunit. 2 of our initiation factors fall off. We hydrolyze that GTP, and we have now completed initiation. And now we have an open site here. So within this large ribosomal subunit, we have 3 sites where tRNA molecules can be. This E site, as soon as a tRNA ends up there, it is ejected, or it exits the machine. This P site, P stands for peptidyl, and it's the site where you have your polypeptide. And then this A site is the amino acyl site, and that's where our new tRNA is going to come in, that the tRNA that has the anticodon to match this codon here. But once that second tRNA comes in, actually once this process is done and we fully attach that 50S subunit, initiation is over and elongation begins. All right, so here is elongation. We've got our second tRNA here came in, had the anticodon that was complementary to the second codon. In this particular example, that amino acid is serine. As soon as it comes in, there's a RNA enzyme here called peptidyltransferase that facilitates forming this peptide bond through carrying out a dehydration or condensation reaction. And at the same time that this amino acid here, methionine, is added to serine and that polypeptide is formed, at that same time the ribosome moves this way. The complex moves down, and then when the complex moves down, this tRNA ends up in the E site and is ejected. This tRNA ends up in the P site and has that polypeptide, and a new tRNA can enter into the A site, and if it has the anticodon complementary to the codon, this reaction will continue. And elongation will continue all the way up until you reach a stop codon. And so, again, on Friday, we'll talk in more detail about all of these processes, but there are at least 3 stop codons, and these stop codons, they don't have a tRNA with an anticodon complementary to them. Instead, they have what are called release factors. This right here is a release factor that has an anticodon complementary to the stop codon. And when this release factor binds, there's nothing for this peptidyltransferase to do in terms of facilitating creating a peptide bond through a dehydration or condensation reaction. And so when that happens, this whole machine is destabilized. This tRNA falls off. The polypeptide is released. The release factor falls off. The large ribosomal subunit separates from the small ribosomal subunit, and they both release this mRNA. That mRNA can be further translated by another ribosome. There may already be another ribosome on this mRNA further down here, further upstream. And when this releases, the polypeptide is released, and then it can go on to be processed. And this is something we will talk more about on Friday as we take this discussion of the central dogma of molecular biology to the next level. So as you prepare for class on Friday, please ensure that you understand the steps of this process of DNA being transcribed into RNA, RNA being translated into protein. Find some questions if you have any, and be ready to go behind the scenes, if you will, in this process.